biology students. Today we'll be talking about genetic recombination as it applies to meiosis. So here's our objective: compare the end products of meiosis and those of mitosis, and we'll describe the inheritable genetic variations that may result from new genetic combinations through meiosis. The three methods of variation during meiosis. So in our previous post video, we discussed crossing over and we discussed independent assortment, and before that we described sexual reproduction. So we're going to try to hit each one of these, and our hope is to get a little bit of quantitative evidence to help us support a claim that that genetic variation can occur in three different ways. Okay, so let's go ahead and get started with, first of all, crossing over. This is a picture that you can remember fairly easily, and for the most part, it does its job. It talks about the crossing over of genetic material. Remember, we're trying to recombine the genetic material, and this can create, of course, a variation. Now, this variation is a material that will be different than the parents, and this helps the organism become more adapted to its environment if need be. Okay, so that is the basic idea of crossing over. Okay, so we're just crossing over genetic information, trying to create that variation. Now, this is probably the one that will probably take a little bit longer to talk about, but we'll talk about it and we'll try to assign some quantitative information to it. But basically, remember, independent assortment is the random separation of homologous chromosomes that occurs during anaphase 1. So it's how those homologous chromosomes are separated that is kind of the point of the concept. Now, independent assortment by itself is not too tough, but the probability is more of what we're concerned about, and how the genetic variation is connected to that. So as a reminder, here's independent assortment. We're talking about, and right now we're in metaphase 1, where this does not show anaphase 1, but it's basically how it lines up. So we can see that we have two chromosomes here, two homologous chromosomes. This would be a tetrad, this would be a tetrad down here. And what we can see here between possibility 1 and possibility 2 is that we have these chromosomes lined up in different spots. Okay, so this is just another variation of what is possible for this particular cell. As they split here, you can see obviously this cell ends up with 2 blue chromosomes. This one ends up with 2 red chromosomes, which eventually get down into the 4 haploid cells that we are describing in the end of meiosis 2. That, of course, create 4 different sperm, or 1 major egg and 3 polar bodies. Possibility 2, same process. The only difference is the red and the blue smaller chromosomes line up in different spots. And so what ends up occurring is we end up with 4 different cells that have different chromosomes. So genetically, these 4 are different than these 4, and all that occurs because of independent assortment. No chromosome is specific to a certain spot. Okay, blue does not always have to line up in that spot. He does not always have to go left, and red does not always have to line up here. She does not always have to go to the right. They can align wherever they want to, so it's a rather random positioning, if you will. So this is independent, meaning that there's nothing that tells blue that they have to be there. There's nothing that tells red that they have to be in a certain spot. It's just simply where the spindle fibers get them to, and how fast they get there, so on and so forth. So really, it's chance. It's really where they line up. So your genetic makeup is really just based on this whole idea of independent assortment. Okay, so here's another way to look at this, and we have this in your notes that you can put together here. But independent assortment is based on probability. So here I have 4 different sets of homologous chromosomes here. We have gray and black, maroon and red, green and baby poop green, and what I would consider a teal and a royal blue here. So each one of these are homologous chromosome pair. Now, as we look at row 1, what is the probability that row 1 lines up like this? What is the probability that black would come first and then gray? Right? So in this case, what ends up occurring is we have 2 different variations here. Okay? 2 different variations. It can be gray-black or black-gray. In this case, we have the same thing. Maroon can line up here, or it can line up over here. Red is also vice versa. So it can go maroon-red or red-maroon. So there are 2 different variations for that. Okay. Next, you can have green or baby poop green, or you could have baby poop green, green here. So there are 2 more variations. And then finally, of course, you can have teal and royal blue, or royal blue and teal in that order. So there are 2 more variations there. Okay. Now, to discuss the probability of the fact that these chromosomes are lining up in this order for this cell, discusses independent assortment. So the probability would be 2 times 2 times 2 times 2 times 2. So it's really 2 to the 4th power, okay? Which turns out to be 16. So this is what this is basically stating. The way that this cell is lined up, this assortment of chromosomes here, there is a 1 in 16 chance that it will align just like that. Just like this, a 1 in 16 chance. And that is only 4 homologous chromosomes right here. There's only 4 homologous chromosomes. Okay? Can you imagine now the probability that a certain chromosome would line up in a certain area when you have 23 homologous chromosomes? So when we look at independent assortment, to summarize it here a little bit, the idea that chromosomes line up in a certain area is false. Each chromosome lines up independently. Like I said, there's variation. There's a 1 in 2 chance that a chromosome will line up in a certain spot. Okay? Now, we can figure out what the probability of a certain alignment will be. In this case, we're using 4 homologous chromosomes to model that. In this case, we found that 2 times 2 times 2 times 2, or 2 to the 4th, gives us a 16 different variation probability. So in this case, 1 in 16 chances of a cell actually lining up like this. And remember, we were only discussing 4 homologous chromosomes. So if we go ahead and make predictions about 23 different chromosomes, how does that get affected? Okay, so probability of repeating the same cell is a 1 in 16 chance based on this 4 homologous chromosome cell. Now, in this case, in a real cell, there are 23 tetrads. Okay, so going back to what we were just talking about as far as probability is concerned, there are 23 tetrads. Now, the number of variations per tetrad is 2. Probability of the same gamete being produced is a 2 in 23 chance. This gives us the idea that not only is it highly unlikely, but there is a 1 in 8,388,608 chance. That is the numbers. That is the quantitative analysis of independent assortment. Okay. Now, assuming that the woman has only 2 children on average. One woman has about 2 children now. Some women will have a lot, some will have 0. So the average is about 2. Now, parents are not likely to have the same child twice. And the reason why is because the likelihood that they will produce the same egg twice, same set of chromosomes, and the man will produce the same sperm twice, and that they will unite are very, very slim. Very, very slim. This is just on a female side. This is a 1 in 8,388,608 chance. Okay? So when we talk about independent assortment, these are the numbers right here. The idea that chromosomes will line up in a certain way and be able to repeat it twice, the probability of that is very, very little. Okay, so people are not likely to have the same child twice. However, when we talk about the combination of sperm and egg, okay, the resultant gametes produced during meiosis are haploid. They create diploid zygotes when they're combined. Now, in this case, the likelihood that the same sperm and egg combination is very, very low. So here's a couple of things. Now, I've given this information to you in your notes. This is from the Cleveland Clinic. It's the female reproductive system itself, and how many eggs does a woman have? Okay. So remember our probability when we create gametes. There's a 1 in 8,388,608 chance that a certain gamete will be produced in a certain way. Okay, so during the fetal life, there are about 6 million to 7 million eggs, and from this time, no new eggs are produced. So females are born with the number of eggs that they're going to be born with. Now, at birth, there are approximately 1 million eggs left, and by the time they reach puberty, only 300,000 remain. Now, of those 300,000, only 300 to 400 will actually be ovulated during a woman's reproductive lifetime. So now you're talking about taking 7 million eggs, which was within the probability of 8,388,608, and now you're reducing that number all the way down to 300. So now the likelihood that we produce the same organism twice is reduced because we only have 300 eggs to work with. So fertility can drop as a woman ages due to decreasing number and the quality of those remaining eggs. So you're really talking about the probability of producing the same child twice directly is related to how many eggs they have left. Now, how many sperm do men have? Okay, how many do they need to just achieve pregnancy? Just one sperm is needed to fertilize an egg and achieve pregnancy. But for each sperm that reaches and fertilizes an egg, there are millions that don't. So on average, there are about 100 million sperm. Okay. Now, in that case, only one is needed. So there are very little that needs to get there. So only one. So the idea here is, why are so many sperm released if it takes only one to make a baby? The idea is to meet the waiting egg. The semen must travel from the vagina to the fallopian tube. So now it's an arduous journey. It's very tough to get there. So very few sperm will actually survive this journey. So for those that complete the trip, the penetration of the egg is far from a sure thing. So not just every sperm gets there, just only a few might get there, and only one will actually make it. So the egg is covered with a thick layer, and it makes fertilization difficult. So many experts, especially those in the evolutionary side of things, will believe that the process may be nature's way of allowing only the healthiest sperm to fertilize the egg, thereby providing the best chance to produce a healthy baby. So at the time of fertilization, a woman has one of her 300 eggs from the original 7 million. So no specific order in which the egg is floated down the fallopian tubes. It's just this one egg. Okay. The man has 1 in 100 million sperm that are used. So there are no specific sperm reaches the egg first. It's just the healthiest will survive. So now you're taking all the numbers that we've discussed. So to produce a sperm twice, it's a 1 in 388,608 chance. And for that one sperm to get there is a very tough and arduous journey. So not all the sperm are even going to make it. And on top of that, of the female eggs, only 300 have survived, and only 1 of 300 have survived. So now the sperm and the egg are coming together. So the probability to have the same individual twice has been greatly reduced based on this whole idea. So random fertilization basically boils down to the idea that of the woman's remaining eggs, only one is present to be fertilized, and of the 100 million sperm that are present, only one is needed to fertilize, and only a few even make the journey from beginning to end. So when we talk about, and I'm just scrolling back here, we talk about these three things all occurring during meiosis. And again, fertilization doesn't occur during meiosis, but it's a direct product of meiosis. Crossing over creates that genetic variation by switching and recombining genetic material. Independent assortment is simply where homologous chromosomes will line up. Okay, and we talked about the probability that the same combination of chromosomes is a 1 in 8,388,608 chance. And random fertilization is talking about the reduction of the number of eggs that are present in a female, and the only a handful of sperm might make it to the egg. Only one of them is actually there. It's not by choice. It's a chance that it gets there. So random fertilization is just referring to those things. So at the end, these three things, as we've discussed, are the three methods of variation of genetic diversity that are caused by meiosis.